So before going to our second channel, the sodium channel, let us look at why do we need it in the first place. Why can the potassium channel, which also transport a monovalent ion, act for sodium ion? Well, before we go into it, let me tell that sodium ion is obviously smaller than potassium ion if you look into the periodic table. But how does the size makes a difference? As you can see here, the potassium ion can interact with all the 8 oxygen atoms properly. Because of its size and size of the pore, it will have equal attraction from all the oxygen atoms. Even when it's outside or even when it's inside, it is stabilized. However, as sodium ion will try to enter, it can interact with only 2 oxygen. Because of this, it will not be stable inside the pore, because it will have unequal amount of force pulling it towards the oxygen. This is why we need a different sodium channel with the proper pore size. And the sodium ion also has some different properties which is useful for different cell function, which will look. The sodium channel, just like potassium channel, is voltage gated, which means it responds to voltage. There are other gating systems also, but here we will talk about the voltage gated sodium channel. Now it also has 4 subunits, each consisting of 6 alpha helices, membrane spanning alpha helices. It also has 2 beta subunits, 2 beta sheets which are called beta 1 and beta 2, and they make the inactivation domain, which was not present in potassium channel. We'll look into its function very soon. Here also the S4 helix act as the voltage sensor. Now we'll look into the classical model of sodium channel functioning. There are different models proposed, but here we'll look at the classical model. This suggests that the S4 domain move in and out of the cell, and here we have the inactivation domain, which will look later. So the S4 alpha helix, the voltage sensor, as usual has its positive charged amino acids. How does it help in this case is similar like the last time. So the outside of the cell is usually more positive than the inside, which gives us a potential difference, usually taken as negative, and the magnitude can depend on the cell type and the environment. Now the positive charge outside repel the S4 alpha helix and it stays towards the inside of this plasma membrane, the inner plasma membrane, the inner part of the plasma membrane. Now what happens when there is a change in the potential, when the outside becomes more negative as compared to inside? This can be happen due to different stimuli. Now this will result in S4 helix being attracted towards the negative charge because it has positive charge, and it will also be repelled by the positive charge that is inside. So this is not actually a positive charge or negative charge, it is just the relative difference in charge. So it will move upward and the pore will open. Now this is the kind of the gate of the pore, and this let the sodium ion enter into the cell. Now this gate or the pore is composed of S5 and S6 helix as usual, and it will also have a ion filter, which will be oxygen atoms arranged in a way so that they will stabilize only sodium ion. Now potassium, because of its larger size, cannot enter, but sodium can. But when sodium enters into the cell, the positive charge inside the cell will increase, and as sodium keep entering, the potential difference will keep rising. Now it will rise and rise till it's reach a threshold. Now what is this threshold? We'll talk later. Now look into something interesting that happens only in sodium channel. Here the inactivation domain comes into play. As the threshold is raised, it blocks the pore, and as the pore is blocked, the sodium ion cannot enter into the cell. The channel is open, but the sodium ion cannot enter because the channel is inactive. It's not closed. Even if it's open, it's inactive, although it's not closed because it cannot be opened. Now we need to restore it to the closed state so that we can reuse the channel, right? We cannot go back to open from inactive as we could in potassium channel. So what happens is, when the potassium channels open somewhere in the cell, the potassium ion will move outside. The potassium ion will make the extracellular site more positive. When the polarization change, the sodium and the sodium channel will again go back to its closed state, as the S4 helix will be repelled inward and the pore will close, removing the inactivation domain. Then again, when there is a potential change, it can open, so it will go back to closed state, and when there is potential difference, it will open again, and then it will be inactivated until it is closed again, but making the outside positive. Now the cell has gained some sodium ion and lost some potassium ion, and it needs to restore it. By restoring, I mean it needs to make the sodium concentration outside the cell more than inside, and similarly it needs to make the potassium concentration inside the cell more than it is outside. Now this is, we have a sodium channel with the blue dot representing a sodium ion. Here you can clearly see the two beta seeds, the beta 1 and beta 2, the yellow ribbon, not the yellow beta seeds that you are seeing if you are familiar with alpha helix and beta seed structures. And this is the top view. You can see the sodium ion and trying to enter into the pore, and this one is from the cockroach, I think, and it was taken from PDB, protein data bank. You can check out more there.